Hi everyone, my name is Miao Xu. So this topic is about the regulation of platelet lifespan. The human body produces approximately 100 billion platelets per day. This is not a small amount. However, their time in the circulation is very brief, up to 10 days in humans and 5 days in mice. This is very different from red blood cells, which can circulate for up to 4 months in the circulation. So we will start with platelet storage and transfusion. Different from red blood cell, platelet cannot tolerate either cold or warm. So the red blood cells can be refrigerated or stored in room temperature. However, platelet cannot be refrigerated. They will lead to a rapid loss of viability. The gold standard of platelet storage is in room temperature, but even at this optimized condition, the platelets can only last for 5 days. The platelet storage lesion or defects involves both activation and apoptosis. Due to the fast clearance in the storage, the multiple hit model was proposed in 1966. The authors argued that an event in vivo would cause some injury to the platelet, which they call a hit. They hypothesized that each platelet would be capable of withstanding a fixed number of hits, and then the platelet will be cleared from the circulation. However, damage to a platelet from involvement in a hemostatic event will reduce its lifespan in vivo, was never demonstrated directly. So, cell intrinsic mechanism may play a central role in the regulation of platelet survival and lifespan. At steady state, only a very small fraction of these are consumed in hemostatic processes. The majority are cleared by the reticular endothelial system, which is RES, in the liver and spleen. Two apoptotic pathways exist in nucleated cells, the intrinsic and extrinsic pathways. The signaling networks and the cellular events that occur during their activation are very complex and differ from cell type to cell type. Here we see in healthy cells, when BH3 is inactive, BCL2 pro-survival proteins such as BCL2, BCL-XL, BCLW, MCL1, and A1 inhibit the pro-death proteins, BAC and VAX. However, when BH3 is activated, the BH3-only proteins sense and transmit pro-death signals to BAC and BAX in response to stress. BAC and BAX cause mitochondrial outer membrane permeabilization, which is also called MOP, leading to cytochrome C release, formation of the apoptosis, and activation of effector caspase 3 and 7, phosphatidylserine, PS, moves from the inner leaflet to the other leaflet of the cell membrane. So PS in nucleated cells is a typical signal for eat-me. With the PS exposure, RES, which is also the reticular endothelial system, will find them and clear the PS positive cells. In platelet, the PS exposure can be reached either by the prothrombinase complex or due to BCL-XL. And also, whether the extrinsic apoptotic pathway in platelet exists or not is unclear up to now. In 2007, it was demonstrated that platelets depend on BCL-XL for survival. Platelet lifespan in mice carrying a homophilic or knockout ally of BCL-X, the gene encoding BCL-XL, will reduce in a dose-dependent manner as shown in the figure. So its role is to restrain the activity of BAC and to a lesser extent BAX. So BAC deficient mice exhibit an almost doubling of platelet lifespan in vivo. In contrast, platelet counts in BAX knockout animals are normal, suggesting BAC is the central factor that limits the lifespan of each individual platelet. So it was proposed that as the platelet circulates, BCL-XL levels decrease until a threshold is reached. The available data support the notion that platelet lifespan is regulated by an internal timing mechanism which dictates time to death, the molecular clock model. So it is currently defined as the time-dependent degradation of BCL-XL, offers one potential explanation for that mechanism. However, there is no definite evidence that the molecular clock model exists, especially represent biological reality. One would expect to find less BCL-XL protein in older platelets. However, as you show in this Western blotting, which is not the case, the protein levels in platelet lysis from wild-type and Bax-deficient mice, and found that the BCL-XL protein levels are very similar in both blotting. This finding raised the question of whether the activation of Bax-mediated platelet apoptosis at steady state is the result of more complex mechanisms. So in 2011, Dr. Hofmeister tried to address this question from another perspective. So the importance of carbohydrate modifications on surface proteins in mediating platelet clearance is increasingly recognized. And in vertebrates, glycosylated surface protein are commonly captured with terminal sialic acid. Removal of sialic acid, which is also called desilylation, exposes the next underlying sugar, galactose. So early studies indicates that loss of sialytic caps on platelet glycoproteins could be a normal occurring process during platelet in vitro and in vivo aging. And studies on platelet desilylation also showed for some immune thrombocytopenia, their platelet lifespan is also affected due to platelet desilylation, the losing of the sialic acid. However, how platelet lifespan is regulated in details is still an open question. So thanks for your attention, everyone.